In this chapter, we'll look at the molecular basis of genetic change. This information is partially in chapter 18, and it also includes some extra information that I've decided to teach that is not included in your textbook. So what we're really going to talk about here is what it actually means when we talk about different alleles such as big A or little a, and what does it actually mean at the DNA level when we talk about dominance or recessiveness. So I'm going to start off by talking about a human genetic disorder, phenylketonuria, or PKU, which is an example we'll use throughout the rest of this video. So people who have PKU, it turns out they are missing a very important enzyme which is required to convert phenylalanine, the amino acid phenylalanine, into the amino acid tyrosine. And so if you eat a lot of protein in your diet, you get a lot of amino acids, and they need to be broken down into a form that we can actually use for energy. If you are defective for the PAH enzyme, phenylalanine will start to accumulate in your body, and it will eventually break down into a toxic byproduct, which is called phenylpyruvic acid. Phenylpyruvic acid can actually cross the blood-brain barrier and damage the brain. And so eventually, if you continue to have high phenylalanine in your diet and you can't break it down, it will permanently damage the brain. Now, if we look at individuals who have PKU, they need to have a mutation in both of their PAH genes. So this is a schematic of the PAH gene, and it has a number of exons, which are these little black parts, and then they're separated by introns, which are numbered here. In individuals with PKU, there are a wide variety of different types of mutations that have been found. So, for example, all these positions on the top here indicate in this particular cohort how many individuals had a mutation in that particular exon. There are also some mutations that have been located in introns, so probably some of those will affect the splicing, the correct splicing of the intron, so that the messenger RNA is not formed properly. So we're going to focus for the rest of this lecture on how we interpret mutations. How do we describe them? And there are a number of different ways that we describe mutations. Sometimes we talk about the specific change that is occurring in the DNA sequence. Another way to think about a mutation is which part of a gene is affected, whether it's an exon, whether it's a promoter, something like that. Another way to think about a mutation is just focusing on the actual result. So how does the change in the DNA sequence affect the function of the product of that gene? And then finally, we're going to talk briefly about dominance and recessiveness and how that allele behaves compared to a different allele. So we're going to start off with this first one, what kind of terminology we use to describe various changes of the DNA sequence. So if we look at a particular protein coding gene, it has a particular sequence, and that sequence will encode amino acids. And so this is the normal version. There are 4 amino acids that are encoded here, and each codon has 3 bases. There are a number of different types of mutations that can happen. If you change some bases, so for example, in this particular case, if we changed this A in the arginine codon to a C, that change does not actually change the amino acid, since there are multiple codons which call for arginine. And so this would be called a synonymous mutation. This will really have no effect at the protein level. You can also get a change that will result in a change in amino acid. And so here we changed an AGA to an AAA, which calls for lysine. Now, we call this a conservative missense mutation, and the reason why we call it a conservative change is that it turns out that arginine is a positively charged amino acid, and lysine also has a positively charged side chain. And so these two amino acids are very very similar to each other, and it's much less likely to cause a significant disruption of the encoded protein. A non-conservative change would be a change to a different type of amino acid. So here we change from an arginine to an isoleucine. Isoleucine is amino acid with a hydrophobic side chain, and so it's much more likely to disrupt the function of that protein or the structure of that protein than a conservative change would be. And finally, we can change a single base, and occasionally that change will result in a new stop codon. So here we've changed from an AAG to TAG, and that single base change introduces a stop codon so that when the ribosome is making that protein, it's going to actually think that's the end of the protein and just halt right there. And so you would actually not make the rest of the protein that follows that sequence, even if there were no mutations in this following part. This is called a nonsense mutation. We can also have mutations that lead to an extra base pair or a deletion of a base pair. Generally speaking, these are called indel mutations, so that stands for insertion deletion. And either one of those two will lead to something called a frameshift mutation. So here we insert an extra base, and what that does is it changes the reading frame of the entire rest of the protein. And so here, instead of reading AAG, you actually have this extra base and we read GAA. And this G that was previously part of this codon is now pushed into the next codon. And so frameshift mutations change the rest of the protein from the place where that insertion or the deletion occurred. Next, we'll talk about another way to define mutations, describing which part of the gene is actually affected. So we'll go over this a lot more in class, but I'd just like to talk briefly about the structure of a gene. And so on the DNA level, you have different parts of a gene, and one thing that we will talk much more about later on in the course is the fact that in eukaryotic genes, you have exons and you have introns. And so the part that is actually transcribed includes both exons and introns, and then once that's transcribed into a pre-RNA, as we call it, the introns will be removed by splicing, and so only the exons will go and be part of the final messenger RNA that will be bound by a ribosome for translation. Now, other important parts of genes include promoters, which are the parts involved in getting that gene transcribed. So if you had a mutation in a promoter, that would likely affect your ability to actually transcribe that gene. We can also talk about exons in different ways. So you'll notice that the RNA, the processed RNA, includes both of these exons in this simple 2-exon gene, but not all parts of the exons are created equal. So some parts of the exon, we know in this particular example that these are really important active sites for the function of that protein. So for example, if we think about the PKU example, the PAH gene, an active site might be the place where you would bind the phenylalanine. And so if you don't have that binding site for phenylalanine, that protein is not going to be able to do its job correctly. Another possible active site in the PAH gene might be a binding site for ATP to actually use energy to accomplish this change from phenylalanine into tyrosine. Next, we're going to talk about some terminology that we use to describe how mutations affect the function of a gene product. So these are terms that are commonly used by geneticists to describe how a mutation will affect the function of a gene. First of all, silent mutation is any mutation that does not actually influence the function of a gene at all. This could be a synonymous mutation that doesn't actually change the amino acid, or it could be something that changes the amino acid, but possibly that change is not so deleterious for the protein, and the protein functions just fine. An amorph is a mutation in which the gene product has completely lost all of its function. And so this is also frequently called a loss of function mutation. That's a little bit more descriptive. Also a null mutation. A hypomorph is a mutation that reduces either the amount or the effectiveness of the gene product. This could happen in quite a few different ways. Possibly you might transcribe less of it than you normally would, or the protein could have some reduced ability to bind its substrate, such as maybe a certain mutation in the PAH gene for PKU would just reduce the affinity of that protein for phenylalanine, for example. It could also reduce the stability of a protein so that the protein just doesn't hang around as long and we don't have as much of it at any given time in the cell. An antimorph is also called a dominant negative mutation, and this is a mutation that has a dominant interfering effect on the normal wild type allele. So usually the effect of an antimorph would be seen in a heterozygous situation where that mutation, that antimorphic mutation, would negatively affect that other normal protein that is also produced in that heterozygote. In class, we're going to spend a little bit more time going over a hypermorph and a neomorph, and I'd like to challenge you guys to just think about what those would be. Lastly, mutations can be defined by how that allele behaves compared to another allele, and that's when we're going to use these Mendelian terms dominant and recessive. So recessive mutations are almost always loss of function mutations or amorphs. And the reason why is because many genes, most genes, are something called haplosufficient. So if we think of the normal dosage of a particular gene in an individual, that normal dosage is 2 copies of that gene. Now, most genes, if you actually lose one of those copies, there is no phenotypic consequence. So that individual who loses one copy is going to look perfectly wild type. And that is actually the definition of this term haplosufficient. Half the normal dose is enough for a normal phenotype. So an example of that is shown here with the tyrosinase enzyme, which if it's homozygous mutant, you are actually albino. Here we have in the nucleus 2 normal tyrosinase genes, and the normal situation is you transcribe both of those genes and you produce protein. So you have this normal dosage which comes from 2 genes of the tyrosinase enzyme. It's required to make melanin, and so you make this normal amount of melanin. It turns out for this particular gene, it is a haplosufficient gene, and so if you have a mutation, an inactivating mutation or an amorph in one of the copies of the tyrosinase gene, you're going to make a mutant or an inactive protein. However, one copy is enough, and so here you have this tyrosinase enzyme. It still is enough to make the normal amount of melanin, and you have a wild type phenotype. It's only when you have a mutation in 2 copies of the tyrosinase gene that you end up with no functioning tyrosinase enzyme and you actually end up being albino. Dominance is a little bit more complicated. And so how is it that you can have a mutation that acts dominantly over the normal wild type allele? One of those mechanisms is for those few genes that are haploinsufficient. So haploinsufficient, just as you would guess from the term, means that half of the normal dose is not enough to give the normal phenotype. And so here, if you only have one of the 2 alleles mutated, it's going to show a mutant phenotype. And because of this, it is going to show a dominant inheritance pattern. And so we're going to call the mutant, in this case, we're going to call it big A. And so big A, or big A little a are going to show the mutant phenotype for a haploinsufficient gene. It's only when you have 2 normal wild type alleles that you are going to show the normal phenotype. Another way that you can have a dominantly inherited mutation is if you have an antimorph. And so again, this is also called a dominant negative. And what this does is it has a dominant interfering effect on the wild type protein. Here's an example of how this can actually happen. A lot of proteins, it turns out, they have to bind into groups, like a tetramer in this case, a group of 4 proteins that all join together to form a functional unit. So this functional unit is demonstrated by this red diamond here. So in order to maybe create a binding site for a substrate, you need to have 4 of them together. So the way an antimorph might function is if this protein needs to form a tetramer, the addition of even one mutant version will destroy that binding site. This mutant protein still will retain its ability to bind to these normal proteins, but it can't actually form the active site. And so what this ends up doing is that mutant will actually poison any wild type proteins and inhibit their function as well. And antimorphs will show dominance over a normal wild type allele. A final way I'll mention that you can have a dominant mutation is a mutation that will affect the ability of a protein to be regulated. So a lot of proteins are regulated by phosphorylation. And so you have a particular amino acid that is required for phosphorylation. And so in this example, maybe you have the active protein is unphosphorylated, and then you phosphorylate it in order to inactivate it. So if you actually mutate that particular amino acid that normally gets phosphorylated, it's possible that it will not be able to be normally phosphorylated, and therefore it's going to be always active. This kind of mutation is often found in cancer, and so we have proteins that will continually induce the cell to keep on dividing.